Hey guys, how's everybody doing? Another day without video. I apologize, but as you can hear, I'm still sick. Not with COVID, fortunately. I had to have that lovely test done, but with bronchitis, and hopefully that will begin to clear up here pretty soon. But regardless, the show must go on, as they say, or in this case, the lectures. So we're going to go ahead with mitosis. Just share my screen. Okay. All right. As always, got to get stuff set up on my end that you can't see. But unfortunately, I can't do this before I start the Zoom thing, which is kind of annoying. But if I do, it makes it go away before I come back to it. Anyway, let me get that. Come on. We're going to go here. Okay. So guys, I'm just going to fill in these letters here for you. We'll talk about some of them in a little bit more detail as we go. And always, if you have questions, let me know. But I'm just going to fill these in without going into a whole lot of detail here. Okay. So there's a lot of vocabulary in this chapter, vocabulary that you'll need to be comfortable with, not just from these lectures, but really for the rest of the course. Okay, let's look at our true or false. The more chromosomes an organism has, the more complex it is. As we'll see on the next page, this is false. 22 of the 23 chromosome pairs in humans are autosomes. This is true. And autosomes are body cells. And then the 23rd pair is their sex chromosomes. All cells in the human body undergo mitosis. This is actually false. Most, but not all. Red blood cells, for example, are not going to go through mitosis. New red blood cells get generated in the bone marrow. Okay, so we get this to move on. There we go. Okay. So now that we're here on this page, I'm going to draw your attention first to, well, actually, I guess I need to go over the vocabulary over here first. Okay, the cell that divides. So when we have our cells getting ready to undergo this process for mitosis, again, mitosis creating two identical cells at the end, right? So as if you were going to divide and you made an exact copy of you, so that there were two of you, exactly the same down to the last detail at the end of your division. So that cell that's going to divide is called the parent cell. Okay. And the two identical cells that come out of it are called daughter cells. You may have noticed that for the vocabulary in this section, we have daughter cells, sorry, sister chromatids. Those terms have nothing to do with sex or gender or anything else. It's just like some people refer to their car or their boat as a she. It actually doesn't have anything to do with anything else aside from just being a name. All right, so let's talk about these chromosomes. Let's talk about diploid number. Di is 2. The diploid number is also referred to as 2n. I'm not going to go into a ton of detail about 2n, but I wanted you to know it in case you see it in your reading, that you'll be familiar with the fact that this is referring to the diploid number. So the diploid number is actually, let's see if I can type this. I make it tinier. I don't think I can. Well, let's find out. The diploid number means that you have sets of each chromosome. Okay. And this means that essentially you've got one set from mom and one set from dad. Okay. And this "and" is important because it's going to help us distinguish between the diploid number and the haploid number. So 2 sets of chromosomes, or 2 sets of each chromosome, I should say, in the nucleus. You get one of those sets from mom, and the other set you get from dad. That then is going to be twice the number that we call the haploid number. Okay, haploid number is only n because it's half of the diploid number. So this means that you have only one set of chromosomes, meaning that you get one set from, you get, actually, it's not even a set, you get each individual chromosome is going to be a different mix. So you get one from mom or dad. You don't have both. So in humans, this diploid number is 46. Half of 46 is 23. So in humans, our diploid number is 23. I'm actually going to pause here before I go over this chart because I want to show you something. Hang on 1 second while I try to pull that up. One second, guys. Thanks for your patience. Okay. There it is. All right. This is a karyotype, which you may recognize as being one of your terms from matching. So this is a human karyotype. It belongs to a human. And actually, I don't like this one as much. What it's going to show is the number of chromosomes that are had. We'll do this one instead. There we go. Okay, let's try that again. All right. So this karyotype, again, human, and if you notice, there are going to be 46 chromosomes. You can count each of them. 1, 2, 3, 4, 5, 6, right? Or you can look at how many pairs there are. There are 23 pairs. Here's the autosomes. Chromosomes 1 through 22 are the autosomes. And chromosome set 23 is the sex chromosome. In this case, this individual is XY, meaning that they are a male. Notice that the Y chromosome is less than half the size of the X chromosome. That's an important thing in genetics because the Y chromosome can't hold as many genes as the X does. Okay, so this karyotype is showing the diploid number because what we see are 46 chromosomes here. Chromosomes are numbered 1, 2, 3, okay, and they're numbered the same whenever you break down a different individual's genetic information and look at their chromosomes, so if you're doing DNA typing for forensics, for paternity, for genealogy, they're always arranged like this. But you'll notice that there are 2 of the number 1 chromosomes, right? Well, that's because one is coming from mom and one's coming from dad. And actually, I'm not sure how these are arranged, if mom's is listed first or not, or if they even know. But you'll see that one is from mom and one is from dad, okay? And it's the same with all of these, okay? Because I've got a situation where I have mom and dad's chromosomes, again, I know I'm looking at the diploid number. If I were to go through and black out one of these for every single chromosome, I'd be left with the haploid number because I'd have only one chromosome for each, okay? And that would be like the mom or dad situation. I also want to point out the X and Y chromosomes. Obviously, we know we get one from mom and one from dad. Well, since females are XX, mom only has an X to give, right? So it is the father with his XY that can give either an X or a Y. So I love that all of the kings in the past centuries who have, had their wives put to death or justified it for divorce, King Henry VIII, for not giving them a son. Well, it was actually totally their fault. They kept providing the X, not the Y. Okay, I just always found that fascinating. How so much of history it talked about women bearing the son for somebody, but really it was the man that passed that along. Okay, so I wanted you to see what a karyotype looked like. Hopefully this can help you envision a little bit more of the 46 versus the 23. What page am I on? No, I'm on this one. Okay. So I gave you the terms. Then if we look over here, again at our chart, we talked about the fact that the number of chromosomes is not a reflection of the complexity of the organism. So I'll show you again. This is a carp. I always think about it as kind of a gross trash fish, I guess because I've always seen it in dirty water. It's a fish, right? Well, its diploid number is 100. It has 100 total chromosomes. Compare that to humans who only have 46. You would think that a human would be more complex than a fish and that therefore it would have more chromosomes. But this isn't the case because again, it's not a reflection of complexity. Then we have a wolf who has 78 chromosomes. Us, as I showed you, 46. A tiger has 38, and a carrot, a guy threw in a carrot, has 18. Notice also that the diploid number is exactly 2 times the haploid number, okay? So if I did 2 times 50, I would get 100. And in this case, is referring to 50. N is 39, N is 19, N is 9, okay? The reason they put an N there is because, as you can see, the number varies between species. So N is just the haploid number of that particular species. Okay, let's come down here to the mitotic spindle. This is sometimes called the spindle apparatus. Just depends on the text that you were looking at. But the mitotic spindle has the function of assembling something called spindle fibers. The spindle fibers are specialized microtubules that are going to attach to the centromeres of chromosomes so that they can be pulled apart during mitosis or meiosis. I'll go ahead and type this for us. Maybe. Okay. Guys, I can't type, I can't spell. It's not really different from normal, so I guess I can't accuse, blame it on being sick. Oh my goodness, I can do this. All right, so what is the mitotic spindle made of? I will tell you. Mitotic spindle is made up of spindle fibers. Spindle fibers are made of specialized microtubules. Okay. So over here, I've tried to point these structures out to you. The mitotic spindle is here at the mitotic poles, and then they are going to kind of assemble these spindle fibers that come out. You'll notice that some come out from here, some come out from here, right? And although you can't necessarily see it, through here are the centromeres of chromosomes that these guys are attached to. So in this image, all these little pink points are the centromeres of the chromosomes, okay? The blue are the chromosomes themselves, the green are the spindle fibers that are attached. This cell is in a stage of mitosis where the spindle fibers are about to shorten and pull these chromosomes apart so that we can have two complete sets in the new cell that's forming. The mitotic spindle, to me, works like a winch and pulley system. Did I spell pulley right? I don't know. So let me show you another image real quick. Some of you guys may be familiar with a winch and pulley. But it kind of, well, it looks like all of these that I'm showing you. Here's one. I'm trying to find, what did I search last time? Oh, this is the one I wanted. Okay. So if this whole system here is the winch, right? All of this. And then this would represent the mitotic spindle, this whole system. And the cable, right? The cable represents the spindle fibers themselves. So just like on this, I'm assuming this is a vehicle, maybe a boat, I can't tell, maybe attached to a truck. The spindle fiber, in this case this cable, is attached to something, okay, that needs to be pulled down, pulled in, pulled closer. When the winch is activated, either electronically or by a crank, this whole thing spins, pulling the cable back in and shortening it. And as it shortens, it pulls in whatever's attached to the other side of this cable. That's the same way that it works in mitosis. The mitotic spindle, okay, is this whole winch system spindle fibers are the cable. The spindle fibers are attaching to the centromeres of chromosomes. And then as the mitotic spindle shortens these, right, pulls the spindle fibers back in, it pulls the chromosomes apart. So there are a lot of actual mechanical processes that happen in the cell to make mitosis possible. Show you this one here too. There's a decent one. It doesn't give too much. Oh, we're just going to go with this one if it clears up. Not going to clear up. This will work. Okay, so right here, this is indicating a centromere, a center point. This is showing a single chromatid, just one here. Up here, you'll notice that these are sister chromatids. So that's, exact copy. This one has made a copy of itself exactly, attached to the centromere. In the Winch and pulley system, mitotic spindle's over here. Just annotate it. So you've got your mitotic spindle, and then you're going to have your spindle fibers that are going to attach to the centromere. And we'll pretend like this is a double chromosome too. Mitotic spindle, spindle fibers, and then these will get pulled apart. Actually, I should have made it here. So that this chromosome gets pulled that way, and this one gets pulled that way, and they end up being separated. Excuse me. Guys, as always, I can't see your face, so I have absolutely no idea if this is confusing the heck out of you or if this helps. So as always, give me your feedback. Let me know if any of this information is helpful, too much, confusing. Yeah. All right. So let's go through and just finish this off. Talking about mitosis. Mitosis is a form of asexual reproduction. In other words, there is no sex that takes place. There is no exchange of gametes, because frankly, that's what sex is. It's an exchange of sperm and eggs, which are known as gametes. So you have this genetic information that gets shared between the two. Mitosis doesn't require that. So it's going to create clones, essentially. It creates two identical daughter cells. And those daughter cells are diploid, has the full number of chromosomes. So you're talking about mitosis in a human, you're going to have 46 chromosomes in the daughter cells. This is different from what happens in meiosis. Let's see if I can hang on 1 second. Okay. Come on, goodness. Purposes of mitosis. All right, so kind of important. Growth. This is having more cells as the organism grows. I'm pretty sure that we all look different than we did when we were 5. Our bodies have changed, our features have changed. Hopefully we're all a little bit taller. That's all because of mitosis. Without that, we would all be the size of babies. It's also to repair damaged cells. Ever had a cut? Ever had a broken bone? Ever had surgery? More than likely, most of those smaller injuries have completely healed to the point that you can't even see a scar anymore. Thank you, mitosis, for that. Imagine what would happen if every single cut, scratch, or injury we've received over our lives just stayed that way in our skin. I mean, technically we wouldn't live long enough because eventually we'd just bleed out from cells that don't ever regrow to cover. But, you know, we don't have all of those marks everywhere, even from something as simple as a skinned knee because of mitosis. New cells grow and cover those up most of the time without even being able to tell that that's where something had happened. Mitosis is also there to replace cells that are lost. I think this is funny because I'm not talking about loss like, oh no, you know, the liver cell wandered off and doesn't know where it is. No, it's cells that have been lost to permanent damage, injury, viruses, cancer, things like that. So lost cells are unfixable cells. Remember that term apoptosis? There are cells that know that they're infected and will purposely destroy themselves to prevent, you know, the infection from being spread to other cells or for the mutation from being replicated. Okay, so we talked about blood cells not always performing mitosis. Actually, I'm not completely sure why that was put in there, because they don't necessarily. But bone cells and skin cells and the cells that allow your hair to grow and your nails, all of these things are constantly doing mitosis. Okay, I'm just going to write down these phases here pretty quick. Just go over some of the highlights. You will need to remember some of your terminology from the cell section. So in this first phase of mitosis, which to remember it in order, just remember PMAT, prophase, metaphase, anaphase, and telophase. Although I always forget that in this class they add prometaphase, so you get PPMAT. In prophase, you have the nuclear envelope dissolve. The chromatin, which is that uncoiled DNA and protein in the nucleus, is going to condense to form those chromosomes that we're used to seeing. And the mitotic spindle forms. We have this kind of intermediate step called prometaphase. In this prometaphase, between prophase and metaphase, the spindle fibers are going to attach to the centromeres. Then we move into metaphase. Chromosomes line up at the cell equator down the middle. They do line up in order to make it possible for the spindle fibers to attach to the centromeres. If they were just a jumble in all different directions, it would be really hard to make sure that every chromosome got pulled apart. Okay. All right. our last page here. So we've gone through prophase, prometaphase, metaphase. Now we're at anaphase. Anaphase, this is where we have the sister chromatids that are actually pulled apart. So the steps before now have been leading up to them pulling apart, but now they're actually going to be separated. And this is where the mitotic spindle, or our winch, is going to pull them apart from each other. Okay. Finally, we have telophase, which is the last step of mitosis. This is usually a reverse of prophase. So if you remember what happens in prophase, just undo that, and you have what happens in telophase. So the spindle fibers detach from the centromeres. The nuclear envelope is going to reform. It reforms over the new genetic material that's been separated and put into new nuclei. The mitotic spindle gets deassembled or broken down. And the chromosomes unwind again back into that chromatin. Okay. Then we have the division of the cytoplasm, which is called cytokinesis. And this is where the cytoplasm itself kind of pinches off to form two new daughter cells. Remember that these daughter cells are identical. Okay. All right. So this is basically mitosis. Let me mention here that cytokinesis can start during late anaphase or telophase. So cytokinesis is actually happening along with the final stages of mitosis. Okay, question of the day. How do cells check themselves before they wreck themselves? I don't know why I had to say it like that, but I felt I needed to say it like that. So this basically is, when we talked about our pre-lecture assignment, we had the G1 checkpoint, a G2 checkpoint, and the mitotic checkpoint. That's how they check themselves. They make sure that, are we really ready to go? Did we really duplicate everything we're supposed to? If the answer is yes, it goes on to interphase, or I'm sorry, S phase. During S phase, we should have DNA replicated. After S phase happens, we get through to the G2 checkpoint, and it's like, okay, did we actually make all the copies here? Are we good? If the answer is yes, we proceed to mitosis. And here it stops and says, do I have all of the necessary organelles, proteins, DNA, etc., for mitosis to continue? And as long as the answer is yes, and we move through these phases and end up back here at a reset point, which is called G0. But you don't really need to, I think, remember that for this. Okay, I get to stop talking now. Fantastic, thanks for hanging in. As always, please let me know if you have questions on this, and I will talk to you soon. Yeah, have a good rest of your day, guys. Bye.